Good morning, and welcome to our special segment on the Minimal Genome Project. What is the Minimal Genome Project, and what are its primary objectives?
The Minimal Genome Project aims to identify and create the smallest set of genes necessary for a cell to function. By doing so, we hope to gain insights into the fundamental principles of life and the basic requirements for cellular survival and replication.
That sounds like a crucial endeavor. Can you explain the significance of stripping down a cell's genome to only essential genes?
By removing non-essential genes, we can isolate the core components responsible for life's basic functions. This not only deepens our understanding of biology but also holds promise for creating custom-designed organisms for various applications.
Impressive. How do scientists go about constructing a minimal genome? What are the key steps involved?
It involves an iterative process of design, build, and test. We start with a full genome and then carefully eliminate genes, observing the impact on the cell's viability. We repeat this cycle until we identify the minimal set of genes required.
I see. Have there been any major breakthroughs in the project so far? Any milestone discoveries?
Absolutely. In 2010, the JCVI SYN 1.0 was created, the first synthetic cell with a minimal genome. More recently, JCVI SYN 3.0 with even fewer genes was developed, marking significant progress in our pursuit of understanding life's essentials.
That's incredible progress. What challenges do researchers face in creating these minimal genomes?
One major challenge is ensuring that the stripped-down genome remains viable and functional. We need to carefully balance the gene removal to maintain cellular integrity and function.
Indeed, maintaining function is critical. Could you elaborate on how the synthetic minimal cells compare to natural cells in terms of growth and evolution?
Surprisingly, the synthetic minimal cells can evolve just as fast as natural cells, showcasing the robustness and adaptability of life, even with a streamlined genome.
That's remarkable. How might this knowledge be applied practically in the future, for example, in medicine or biotechnology?
The minimal genome research holds immense potential for creating tailor-made microorganisms for various applications, such as producing biofuels, pharmaceuticals, and other biotechnological advancements.
So, these synthetic cells could be harnessed to serve humanity in various ways.
Indeed, they offer great promise in revolutionizing industries and addressing real-world challenges with precision and efficiency.
Excellent. Now, let's delve into the technical aspects. What techniques are used to synthesize these minimal genomes?
We use advanced genetic engineering techniques, such as CRISPR-Cas9, to precisely manipulate and edit the genomes of cells.
Speaking of precision, how accurate and reliable are the outcomes of these synthetic minimal cells?
The outcomes are subject to rigorous testing and validation to ensure accuracy and reliability, and the results are continuously refined and improved over time.
It sounds like a meticulous process. Are there any ethical concerns associated with creating synthetic life forms in the lab?
Indeed, ethical considerations are paramount. As scientists, we are committed to conducting responsible research, and the ethical implications of synthetic biology are continuously discussed and evaluated.
Responsible research is essential. Are there any particular environmental or safety concerns when working with these synthetic cells?
Safety protocols are stringent to prevent any accidental release of synthetic organisms into the environment. We work in controlled environments to mitigate potential risks.
That's reassuring. Can you tell us about any ongoing research or future directions in the Minimal Genome Project?
The field is rapidly evolving. Ongoing research aims to further reduce genome size, explore more complex organisms, and gain deeper insights into the principles of life.
Exciting times ahead. Finally, what do you envision as the ultimate goal and impact of the Minimal Genome Project in the long term?
The ultimate goal is to unlock the fundamental mysteries of life and use this knowledge to develop innovative solutions for various sectors, benefiting humanity and the environment alike.
Thank you so much for your time and invaluable insights. It's been a pleasure to learn from you today.
You're welcome. Thank you for having me. It was a pleasure to be part of this discussion on the exciting world of minimal genomes.